Club. Going to introduce Dr. Andrew Ramstead. He was one of the very early phage hunters. There were pictures of him up in Sandy Wardell's classroom. And in that visit, they discovered tennis Wardell, the phage tennis Wardell. They named it in honor of their teachers, of course. They're awesome students. And he spent a couple years studying tennis Wardell, including the effect on biofilm formation. And I don't know how much he'll tell you about that, but those were hard experiments because he had to establish the conditions, the liquid conditions on which SMEG would grow in a biofilm, et cetera, and see the effects. So I'll leave you with Dr. Andrew Ramstead.
So, hello, my name's Andrew. I'm a postdoc at the University of Utah, and today I am going to give you, I guess, a brief summary of my scientific journey, the role that the phage digging program at Montana Tech played in that, and why I think programs like Phages is so important. So, when you're in secondary school, you're an undergraduate in college, you take a lot of lab-based courses, and they prepare you in some ways, but not in others. So where are they useful? Teaching you to do careful experiments. You know that the experiment should work, that what the results should be, and if you don't get that, that probably means you didn't necessarily do something right, and maybe you need to be more careful in the future. It helps get you comfortable around a laboratory and exposes you to basic techniques and instruments. But where it's less useful is a lot of conditions that occur when you start doing actual science. What do you do when the experiment doesn't work? How do you troubleshoot? What happens when you get an unexpected result? Where do you go from there? Where do I even find clues as to what is happening and what to do next? And a lot of the process of becoming self-sufficient in science can be a very steep learning curve that for most people begins as a graduate student. And so in an ideal situation, for first couple of years, you get a lot of supervision, and they kind of hold your hand and they give you a lot of advice, and you become more and more confident. And then as you progress through your graduate career, you become more and more independent, so that by the time you graduate, you become the expert in your project. Nobody understands your project better than you. So that once you now enter the world of becoming a postdoctoral researcher, you're pretty self-sufficient, and you can go to labs, and the professors will give you projects, and they can feel confident that you'll make some project without them being over your shoulder, some progress without you being over your shoulder the entire time. But this doesn't always happen. I've seen a lot of graduate students over my time, and it really depends on the lab. Some labs, the professor is just too busy with administrative stuff, or the lab's too big to really give the graduate student much attention and give them that initial help that they need. On the other end of the spectrum, the lab might be in a very competitive field. They might be running out of money and need to get new grants funded, and so the professor cannot wait for the student to figure it out and to kind of go on a scientific journey. They need good results right now. And so I found a lot of graduate students either have their hand held and dragged up the hill, or they make about halfway up and then crash and burn. And so what programs like Phages can do is they can flatten the learning curve by helping you get through some of those early difficulties and working out some of the bugs, either secondary school, undergraduate, in a hopefully less stressful environment where education and training is the primary purpose. So that by the time you get to becoming a graduate student, now you can hit the ground running and you are less dependent upon your environment. And of all the graduate students I've been exposed to over my years, you can really tell the difference between those that had extensive experience with research before going to graduate school and those that didn't. It makes a big difference if you want to go into academia. So another analogy, courtesy of aqua blue pools. I just found this image online. Phages can serve as the shallow end of the pool where you can get your feet wet, you can splash around a bit, you can learn how to swim so that when you go and you get to the deeper end, hopefully you do not drown. So where did my learning curve begin? So I attended Montana Tech as a chemistry student from 2004 to 2008. And around 2005, 2006, I believe I was a sophomore. I wanted to do more than just classes. I wanted to do undergraduate research. And the chemistry department had some great labs that offered undergraduate research, but they were either very full or they wanted more senior students, and for good reason. But I was impatient, and fortunately for me, at around this time, there was a new professor in the biology department that was getting her lab started, and she was looking for students to help hold tubes during demonstrations, and maybe a few other things as well. So here I found some old pictures of me in the lab. This was probably shortly after I joined the lab. I had this very bewildered look on my face, and I have a picture of how do you streak out bacteria onto an agar plate here. Here's me at one of those outreach visits, doing what I do best, hold the tube. And here on the right, I'm in front of an ultracentrifuge trying to purify some phage. And a couple things I noticed from this picture. Apparently back then I had a go-to hat. And I could have sworn I took my backpack off in the lab sometimes, but apparently not when the camera was around. And I got to learn a lot of lessons from my time doing undergraduate research in the lab. Main lesson, failure is always an option. And it's oftentimes the most common option. So, like many of you here, I also did not find my own phage. I had to adopt one. Data takes you in strange directions. So one of the biggest difference between being a scientist and being an engineer, engineers can kind of control the environment that they work in. Scientists, we're trying to figure out how nature works, and nature doesn't always want to go where you want it to go. You gotta be willing to sometimes pivot and go where the data takes you. Most interesting observations are almost always come by accident. There is sort of a joke that I've encountered in laboratories that figure 5 of the paper was almost always the first experiment that you did. And then you have to go and do the earlier experiments and pretended that you planned it that way. And it's always good to make connections with other researchers. And that's another thing that a program like Phages provides. You all are making connections with each other, with researchers, and you never know when those might come in handy later. So here's Mycobacteriophage tennis wardell that I adopted. As Marissa said, that it was discovered in the classroom of Sandy Wardell and named after her and another teacher. And we did a lot of work with it. We found that it was a lysogenic phage. So 2 major life cycles of phages: lytic cycle, infects the bacterium, integrates DNA into the genome, makes more viruses, kills the cell, viruses are released, rinse and repeat. But it can also undergo a lysogenic cycle where the DNA gets incorporated into the host DNA. It kind of lies dormant and replicates with the bacteria, and then occasionally becomes reactivated and makes more phage. And so one of the hallmarks of a lysogen is they cannot be infected by the same phage twice. So on the left here we have tennis wardell lysogen, on the right wild type SMEG. You see that the lysogen cannot be infected by tennis wardell or supernatant from the lysogen, but could be infected by another bacteriophage we had in the lab, guapo, while the wild type SMEG can be infected by tennis wardell, guapo, and the supernatant from the lysogen. So We found that this was a lysogenic phage, and I was doing a lot of work with the lysogen, growth curves, those type of things. And then one day, I'm pretty sure it was a Friday, I had grown up some lysogen, I had grown up some wild type bacteria. I just left it on the bench. I'll take care of it on Monday, and I left for the weekend. I came back, and we noticed something weird. As you can see, the lysogen here on the left had formed a biofilm, while the wild type Mycobacterium smegmatis did not. This was a completely accidental observation that basically drove the last year and a half of my time at Montana Tech. And I'm not going to go over everything we did, but I went down a lot of tangents to try and figure out why was this happening. Could it be phage integration into an inhibitor of biofilm formation? So we got to sequence the virus and identify the ATP and integrase site, which had homology to another phage. And so we kind of know, or we had a pretty good idea where it integrated. But based off of what was known in the literature about GROL1, this didn't really make sense for what we observed. So we tried some other questions. Was there a difference in their ability to interact with glass? So I found this old picture. I ended up doing these experiments comparing glass Petri dishes to polystyrene. And according to a note I found, sometimes there was a difference and sometimes there wasn't. So it wasn't very conclusive. Were there differences in bacterial protein expression in response to infection? I got to go to Montana State and do these really cool two-dimensional gel electrophoresis assays. And so each one of these dots is a protein. And the darker the dot, the more protein. And we identified dots that were definitely different in the lysogen compared to the wild type. What those dots are, don't know. Weren't able to successfully identify them. Do they have anything to do with biofilm formation? Again, no idea. Differences in cell motility. This is kind of an interesting experiment. We wanted to see the ability for the bacteria to spread across an agar plate from a fixed point. And so we had wild type SMEG, we had a couple of other lysogens, Shabima and Dumbledore, and then we had Tennis Wardell. And Tennis Wardell lysogen likes to stay where it's at. It doesn't like to spread. So this was kind of a cool finding towards the end of my time there. Does this have anything to do with the biofilm formation? Possibly, but I don't know. And so in conclusion, I had no conclusions. I could not find a conclusive answer for why Tennis Wardell lysogen behaved the way it did. I was not ready at that time to be able to answer that question. I was still early in my scientific journey. However, I got to do a lot of things. I got to troubleshoot, expand my mind and my knowledge, learn how to do research-relevant literature searches, one of the most important skills you need as a scientist. Try, fail, and try again. I got to experience being a largely independent, and attempting to be self-sufficient scientist as an undergraduate, which was vital for the rest of my career. I ultimately made the decision to go to graduate school. That wasn't the original plan. I was originally planning on taking my chemistry degree and going to work, but I changed my mind. And this goes back to what I said about connections. I got to make some connections with the Montana State Department of Veterinary Molecular Biology, which is what it was known at the time. I think it's undergone 4 name changes since then. But this was Dr. Ivanka Voyich-Kane. She studies antibiotic-resistant Staph aureus. And she was interested in finding some phages that could infect MRSA. And so she would come to Montana Tech and do some lab meetings with us and talk with us, and I got to know her a bit, and I got to know the department a bit, and it sounded very interesting, and so I ended up joining that department as a graduate student. So continuing on my journey at Montana State now, I went and I did some rotations, and I ended up in the laboratory of Dr. Mark Judala, who is an immunologist, and I've been an immunologist ever since. So it's funny when you all talk about this very interesting phage therapy. Me as the immunologist now thinks, how long do you have before the immune system kicks that virus out? How long is that therapeutic window? Maybe it's longer than I would expect. But I think it'll be a very interesting question that needs to be addressed as people explore phage therapy more. But Mark was a great mentor. He's a great teacher. He really made sure his students got into the literature and was up to date on what was going on in immunology, and sent us to big immunology conferences like AAI so we could get a broad view of what everybody was doing. Sometimes he would come into the lab and give his graduate students pop quizzes to make sure that we were learning what we needed to learn. And so he made sure his students weren't being left behind in terms of their skills and their knowledge. So what did I do there? We were part of a collaborative research effort that was part of the National Center of Complementary and Alternative Medicines. And so there were a few labs at Montana State where we would take nutritional supplements that have or been proposed to have health benefits, things like fireweed and acai powder, and chemists in the Quinn lab would isolate all these molecules, polysaccharides, tannins, all these different things, and then they would give them to us, and we'd throw them on immune cells from mice or cattle or graduate students who were looking to make $20 with donating blood, and see what happens. And so my main contribution to the lab is I developed this in vitro in a 96-well plate antigen-independent immune priming model where I would isolate out the immune cells from the blood, put them on a plate, introduce the compound of interest to them, let them interact for a little bit, then I would wash away the compound and then treat them with a secondary immune stimulant, usually a low dose. And I'm basically asking the question, does exposure to this compound influence the way the immune cells behave? Does it enhance their function, reduce it, do nothing? And I would find data like this. So this is ONO-thean B. This is a alagitanin isolated from fireweed. And I show that pre-treatment with ONO-thean B can dramatically increase the production of this molecule interferon gamma, which is an important effector molecule of the immune system. It has antiviral properties, antibacterial properties, even anti-tumor properties. And pre-treatment of these immune cells with ONO-thean B could enhance the production of interferon gamma to this molecule here, IL-18, which is a danger signal that cells put out. And I can figure out what cells were producing this interferon gamma. So in this case, I depleted NK cells, which express this molecule CD35, and you can see the interferon gamma production completely goes away. So from this experiment, you can kind of conclude that ONO-thean B primes natural killer cells to respond more robustly to IL-18. And I did this with a large number of different compounds and secondary immune stimulants, IL-18, tumor cells, bacterial cells, a lot of different things, a lot of different compounds under a lot of different conditions. And this resulted in a few publications, first author publications, and ultimately it resulted in me getting my PhD. And so made it through the program and got my PhD. And then I moved on to the next step of my career, where I'm currently at. I'm at the University of Utah. This is where I work. This is the Huntsman Cancer Institute. It's half a hospital, half a research institute. Originally, mainly like cancer genomics type research, but because immunotherapy is becoming such a hot topic now in the cancer world, they've recruited a bunch of immunologists there. And so this is where I work, and ended up in the laboratory, Dr. Ryan O'Connell, and what he studied in his lab was microRNAs. So if you remember your basic cell biology, DNA gets transcribed into RNA, and then RNA gets translated into protein. But what if I told you is that there is a very large family of RNAs that never make protein? They actually do their function as RNA. And microRNAs are part of that family. And basically what they do is they bind to the mRNA, the protein coding mRNA, and they regulate the translation of the protein from that mRNA. And it's not an off switch. It doesn't completely shut it down. It's more like a thermostat. It's a minor regulator of protein expression. And it will do this to a bunch of different mRNAs, which code for a bunch of different proteins. But just because it's a minor regulator doesn't mean it's not really important. Because if we knock out one of these microRNAs, miR-146A, from mice, the mice die faster. So here's female, male combined, percent survival over the course of a year and a half. You can see that pretty much all the wild type mice are still alive, but a large percentage of the 146A knockout mice die, especially in the female cohort. We found that this was largely an autoimmune driven response. And to kind of show this, we could rescue this survival, not entirely, but for the most part, here in the gray, by knocking out another microRNA, microRNA 155, just in one type of immune cell. So knock out one microRNA, increase death, knock out a second one, and you can extend their lifespan just knocking out one microRNA in one type of immune cell. And this is one of the experiments we did in this paper. It's kind of a complicated one, but I throw it up because I think it's really cool. It's one of the coolest experiments I was ever a part of. This is a Western blot. So what we did is we took healthy mouse brain tissue and we ground them up and we extracted the proteins and we ran them on an STS page gel to separate them out by size, and we transferred them to a membrane. And then we washed those membranes with serum from these different mice. Well, wild type, knockout, double knockout. And basically what we're doing is we're looking for antibodies in the serum that actually bind healthy tissue protein. And so wild type serum, good. No antibodies binding to your brain. That's what you want. 146A knockout, there's clearly a problem here that we can largely rescue by knocking out the miR-155. And we did this with different organs, liver, kidney, spleen, very similar results. Brain was just the cleanest and most dramatic. And we were kind of shocked. We kind of thought that the brain, we kind of threw this in there. We didn't think we'd ever see antibodies to the brain. So, so that's the kind of stuff I did in the O'Connell lab. I did get a couple of publications in that lab, and it was a great experience. I loved working there. Unfortunately, as is very often the case, the money ran out and grants ran out. Couldn't keep me around anymore doing the projects that I was working with, so I had to try and move on to a new position. And so I went right down the hall to Dr. Matthew Williams' lab, which is where I'm at now. And just to give you just a little idea of what I'm doing currently, what we study in the Williams lab is an immune cell called T cells. And these T cells have a very unique receptor called the TCR, T cell receptor, very original name. But this receptor is interesting because it's formed through a randomized recombination of the genome. And this recombination at the junction points leads to base pairs, DNA-based base pairs being randomly added or removed or mutated. And this leads to a crazy amount of biological diversity in the T cell. So one TCR, one T cell, one TCR. And I saw one paper that suggested that within an adult human body, you have 10 to the 10th different clones with different TCRs in your body. And so in theory, whatever you get exposed to, whatever pathogen, whatever foreign entity you get exposed to, as long as it's a peptide, a protein, there's probably a T cell in your body that recognizes a piece of that protein. And when it recognizes it, these cells become activated and they proliferate like crazy. And they do this, and then they go and they help clear the infection. And then once the job is done, once the infection is cleared, the vast majority of these cells die. About 90% of them die. But not all of them do. There's a subset called memory T cells that last for a long time. And if you ever get exposed to that same pathogen again, or something close enough to it, these cells are ready and waiting to take off again and will actually prevent you from getting reinfected. And so these memory T cells are critical for the development and the effectiveness of vaccines, long-lived anti-tumor immunity. During COVID, they discovered that a subset of people actually had memory T cells that they probably generated from common cold coronaviruses that were actually cross-reactive to COVID-19 and protected these people from getting severe infection. And so these are very important cell type. And one of the questions we asked main question we asked in the Williams lab is, why these cells? Why do 90% of these cells die, but these cells decide to stick around? And so one of the ways we've been addressing this question recently is a collaboration with the Evavold lab, and they have this very cool tool, force probe, where they're able to grab, this is like a vacuum system, grab a T cell, this is a red blood cell, with a bead that has the target that any given TCR, the TCR we want to study binds to. And then they can go and they can push the cells together and then pull them apart at different forces. And they can see how long that interaction is before they become pulled apart. And this is the kind of data you get. So different forces, bond lifetime, and you can just kind of see here with this representative is that different TCRs have different bond lifetimes. And if we follow an infection, so this is a model infection, LCMV virus, and we start with T cells that we know are specific to LCMV, and we start before they were activated, and then as they start to proliferate, and then when they reach peak effector response, right before they start to die, and then memory, and then recall, we can see that the peak bond lifetime changes dynamically. So it starts out around here, drops at day 3. We don't know why it does that yet. Then by day 8, peak effector, it's higher, memory starts going down lower, and the actual, the true memory cells, the cells that are able to respond to a secondary infection, have even lower peak bond lifetime than that. So we can then do a bunch of sequencing and identify TCRs that were either effector biased or memory biased, meaning highly prevalent at the effector time point, but disappear over time, or ones that kind of start slowly. They're not very common at day 8, but they stick around for a long time. And as you can just kind of see here, it's very clear that high bond lifetime by the TCRs leads to short-lived effector cells, while the memory cells had this very low bond lifetime. So very kind of cool biophysics, and we're following up on it. What I'm doing now is I'm making Frankenstein TCRs, where I cut and paste like different key elements of this TCR to see which ones lead to these longer bond lifetimes or shorter bond lifetimes. And ultimately, I'm going to test to see if I can change the behavior of these immune cells just by cutting and pasting small parts of the TCR. So that's what I do now. And so this was my growth curve, starting with why does the biofilm form in the lysogen, all the way to trying to manipulate immune cells with genome engineering. And I can guarantee you that without this would have been much harder. And so I'm a huge proponent of undergraduates getting into the lab, students getting into the lab, and experiencing this before they have to go on and go off the deep end and experience some of the other stuff that they'll experience later on in their career. It's really important. And so acknowledgements, I threw a bunch of names up here. I've had a bunch of mentors and important people throughout my career. But for this meeting, especially got to give a shout out to Montana Tech. I'm really glad I chose Montana Tech to do my undergraduate research. I really enjoyed my time there, both as a researcher and just as a student. I actually think it was a really great experience as an undergraduate student as well. And then, of course, Marissa she took a chance on me as a young undergrad, and she spent a lot of time and a lot of resources into my training and development. And I hope it was worth it, and I hope it continues to be worth it. And whoops. But it made a huge difference in my life, and so thank you very much for that. And with that, I'll be happy to take any questions. Yep.
With that little device, can you tell cross-reactivity between different antigens?
In theory, yes.